It's like Rocio's recordings. All right, recording as well. Okay, so I'll do just a brief intro here, and then I'll turn it over to you. So, yeah. So hey everybody, thanks for coming today. So our speaker today is David Simelane. Am I pronouncing your name correctly?
Yes,
I'm sorry.
correct.
Okay, Simelane, and so he's talking to us from Pretoria, South Africa. So he's a South African. He did his PhD at the University of Cape Town, received that in 2006. Since 1998, he's worked at the Agricultural Research Council, first as a researcher and then senior researcher, and now as a team leader and specialist researcher working on biocontrol of invasive plants from South America and Mexico and Central America. So he's published several dozen peer-reviewed articles and has spoken at a variety of different national and international conferences and workshops. And so I think it should be a really interesting talk, and thanks for coming, David, and I will not take up any more of your time, so you can go ahead and get started.
Yes. Thank you, ladies and gentlemen. I would say good evening if you happen to be on the same longitude as me. But those who are in the far west, maybe good morning. Thanks to the organizers for giving me this opportunity to present this talk today. So I will be talking about mainly biological control, but these are the few things that I want to touch on before getting into biological control of Cestrum in South Africa. I will touch on the invasive Cestrum species that we have here and their distribution, their impact on the environment and also on livestock, their spreading mechanism, how they spread, and then I will go on into biological control, what we have done so far as far as biological control is concerned, and then I will summarize. First of all, invasive Cestrum species, we have got three invasive ones. The first one that we have is Cestrum laevigatum. This one is mainly located along the eastern part of the country, in the eastern provinces. That is the main one. And then we have the one that has only became invasive in recent years. That is Cestrum parqui, and it's usually found in the inland. And then we have the third one, Cestrum orienticum. This one is very stubborn. It's still isolated in an area. It's a town close to Durban. It's called Pietermaritzburg. We've been trying hard to remove it, eradicate it, but it keeps coming back. Those are the three that we have started a biocontrol program against. Generally, all Cestrum species are very, very toxic to livestock. This is very true in South Africa because we've got long dry period. And during those long period of dryness, when grass is not sufficient, animals tend to take foliage of plants, and Cestrum is taken. And if it's taken by livestock in large quantities, that means death to those animals. So that's the problem we have. Not only livestock, but humans as well are also vulnerable. Pigs are also vulnerable. Cestrum laevigatum, especially the one that I said is very, very prevalent in the eastern part of the country. This one, it invades natural forests and pastures. And in the natural forest is a problem because, especially when it is not flowering, it mingles with other plants. So it's easy for the animals just to feed on it. The other one, Cestrum parqui, which is very prevalent around here in Pretoria, where I am, in the inland. This one, it's a headache. It is invading pasture land where cattle are grazing. So it is also very, very problematic of late. Why are these things spreading so fast? Naturally, we know that they are spread by birds and floods. But in South Africa, the spread is also man-made. It's also, pushed by human beings. They tend to grow this cestrum in their own property as ornamental plants, as you can see there. They cut it nicely, they shape it nicely, and this is illegal because cestrum is categorized as category 1 plant according to our NEMBA Act. The NEMBA Act is National Environment and Biodiversity Act in South Africa. So, which means if you are found having this kind of a plant in your yard, that's a criminal offense. But people still continue to have them in their yard. And again, there are some illegal butterfly gardens, which we have clamped down of late, but we assume that they also contributed to the spread because these gardens are illegal and they are not quarantine like kind of structures. So anything can happen. The plants can escape from there and into the field. So these are the things that are causing the spread of these plants in South Africa. And we have two that we managed to contain. We have a program called early detection, which helps in kind of destroying the plants when they're still at an early stage of invasion. So we have managed to eradicate these two. Then biological control. We initiated biological control in 2007, but due to lack of manpower, somewhere in 2008, we put the whole program on hold. But because it was a problem, when I got another opportunity to work on another weed called balloon vine, Cestrum grandiflorum, which is based, whose native range is Argentina and South America. As I was doing surveys on balloon vine there, I keep finding Cestrum. And opportunistically, I started looking closely on insects that are found on Cestrum. And opportunistically, we brought an insect on Cestrum. But I must say that some of the insects that we have found on Cestrum include the butterfly. It's a great species, which we have tried to breed in the lab, but unfortunately, we were not successful. We have also tried to breed another flea beetle. It's a very, very small beetle here that feeds on the leaves. And it's when it lays on the soil and the larva feeds on the roots. That one we have also not been able to maceriate under lab conditions. However, a very small flea beetle called Epitrix, this one we managed to breed it, and we managed to do other tests to determine its safety as a biocontrol agent in South Africa. So we've done some studies on this particular Epitrix. What we have done, we have investigated its biology, looking at its life cycle, the number of eggs it lays during its lifetime, etc. So these are some of the data that is associated with this biology. So in short, in 35 to 43 days, it completes its life cycle. And within a year, it can make 8 to 10 generations, but under laboratory conditions. So We have done host specificity tests. Host specificity tests, these are the tests that we conduct to determine whether the insect is safe as a biocontrol agent. So if you expose an insect to no choice test, it means you are confining it with a plant and without giving it any other choice. We want to see if the insect can feed on that particular plant. If it can feed on that particular plant, then chances are it may probably feed on it even under field conditions. And then we also do multi-choice tests where we put a number of plants together to see if an insect can make a choice. So we have done these 2 studies. This is how we set up no choice tests. There's a single plant species in each of these little cages. So in each, you confine the insect. You confine the insect with a plant. And after some days, maybe 30 days or so, you remove the adults and you see whether the insect has laid on the plant, and also to see if there are some immature stages developing on the leaves. As I said, maybe I should have emphasized that the beetle lays its egg in the leaf, on the leaf, and then the larva develops inside the leaf. And then that's what it does. So the results of the no choice tests, this is what we got. We have tested over 50 plant species. Most of these plant species are in the family Solanaceae. I would say more than 50% of them were under the family Solanaceae. And this particular flea beetle has only developed on 3 species, which is the 3 Cestrum species that are targeted for biocontrol. But in addition, it has also fed on Datura, which is also in the family Solanaceae. But this particular plant is also a weed in South Africa. So its attack by this flea beetle is not a problem for us because it is also a weed. So these are the results of the no choice tests. I had to, because I had a lot of students helping me around here, we had to do a multi-choice test. But when the results are this, we don't really need a multi-choice test because this is a clear-cut result, which shows that the insect is very specific to Cestrum. From here, you can just release the insect. You can apply for permission to release. But for the sake of the fact that we had enough resources, manpower to do the multi-choice, we had to do this, putting in a number of plant species together with the target plants, together with the Cestrum species, to see if the insect can make a choice. And again, we found very, very similar results. This time, Datura was there among the group of plants. It was not selected at all because the insect had a choice to feed, to select between the Cestrum. So it was totally ignored there. So the only 4 plants that were, the only 3 plant species that were fed on were the Cestrum species. So in summary, what does the whole thing mean? It means that Epitrix species that we have just tested is adequately host specific to be released as a biocontrol agent for Cestrum in South Africa. So from now, I will be writing a release application, which we submit to the Department of Environmental Affairs and the Department of Agriculture. And it's a long report. We call it a risk analysis that we submit to the department. And then they are the ones who give us permission. They review your application and then they give you permission to release the insect. We hope we will get the permission. However, as you know, that Epitrix is also among a group of species that are known to be pests. They are known to be pests of potatoes and other solanaceous crop species. So in that way, my next step now is to make sure I get the full name of Epitrix. We now know that it is a new species. So I've got some scientists to help me describe this. We'll do some morphological and molecular description on it, and then we'll describe it and come up with a name. But we don't believe that Epitrix alone will be a silver bullet. There's still a lot that needs to be done in terms of bringing in or adding new natural enemies into the country and test to add on to the Epitrix. So we had a plan to go to South America 2 years ago, but unfortunately COVID came. We had to postpone that. But hopefully next year, March, we are going back to South America, Brazil in particular, and Argentina, to look for more natural enemies. We will target, now that we have a leaf feeding agent, we'll try to look for flower and seed feeders and also stem feeders, because we also want to avoid some kind of competition. You have to mix the insect and have the insects that will attack different niches of the plant. So that will be done. So I think that is where I can end. I would very much like to thank the people that have been helping us with the tests and everything, and also Environmental Affairs for funding the project, and IBONE, which is an institution in Argentina that works with us there, and NRF for also partially funding the project. I think that is the end. Thank you very much.
Awesome.
Yes.
Thank you, David. It sounds like I'm getting a little bit of feedback off of a microphone, but yeah, okay, so that's interesting. We don't hear a lot about about Solanaceae as invasive species, so it's interesting to hear that side of things and what's going on. So normally we just do a question and answer session, and people can either type question in the chat, just say question, and then you can ask a question, turn on your video and ask, or if you want to type out the whole question, you can feel free to do that too, and I can read it. Yeah, so just if anybody has any questions, let us know. Give people a second to think about it.
Hey, yeah, I got a quick question.
Yeah, go for it.
Great talk, by the way. Thank you. When you did your surveys and looking at Cestrum in its native range, did you ever see like hornworms eating on them?
Did I see what? Can you repeat that?
Hornworms? They're like big caterpillars with a horn.
Hornworms. Okay,
Yeah.
okay. All we saw was a worm, and we brought it in here. It was a green worm, and only to find that it was a greater species. These are called white, glass-winged butterflies. The wings are transparent. So that is the one that we have found so far, the Lepidopteran that we have found. Okay. We found it in Argentina, yes. But when we brought in here, when we tried to breed it a number of times, we could not succeed. It didn't do well. The populations dwindled until it disappeared.
Interesting. Thank you.
Okay, thanks.
Anybody else? Any questions? I have a question. I always kind of wonder with the biocontrol setups, and I don't know that much about it, but so like with the beetle, do you ever worry about sort of the beetle evolving new host specificity as it's introduced into a new ecosystem? And so my follow-up to that is there some sort of like, what's the procedure for like continued monitoring after when you release the biocontrol agents? Does that question make sense?
Okay, I don't know. I'm not getting it clearly. So what's your concern is when we release the agent, do you think that it will evolve and become something else?
Oh, well, so it's, yeah, so it's introduced and there are maybe other Solanaceae, native Solanaceae species, for example, and, they're insects and they have short generation time. So potentially evolution could be happening over a fairly short time scale. So yeah, my question is if that's a concern or if there's monitoring for the specificity going forward in time.
Yes. Let me say it clearly that when I say challenges in biocontrol, I mean challenges in the sense that these weeds are among the plant species that are crops. So it's always difficult to find very specific agent for Solanaceae because the agent must be very, very specific. It should not attack any other Solanaceae. So yes, the tests are very rigorous. You repeat and repeat and repeat many replications to make sure that it really it's very specific to Cestrum. So great care is taken to make sure that the insect is very specific. It cannot feed on any other Solanaceae.
Cool. Okay.
So by the time we release, we're very sure. We're very sure that it's not going to take other Solanaceae plants.
All right. Thanks.
It's a process that takes years. We started working on this on 2015. Now it's 2023. So it takes a long time to get it done, to make sure that it is safe.
Awesome. Thank you.
Yeah.
Looks like we have another question from Deidre. I don't think we haven't had too many questions so far. So if you want to go ahead and ask your question.
Yes.
Yeah, so I'm really sorry I missed the first part of your talk. What I did see was really interesting. I had a question about the relationship between more of the no choice and the choice assays. So it seems like when the Epitrix have a choice, they really just avoid Datura. But were the ones in the no choice assays that do some damage on Datura are, can you rear those out at all on Datura, or all of them fail to complete development?
Okay. First of all, the damage on Datura was just feeding damage. It did not lay, it did not develop on it in the no choice test. Okay. And that is very normal for insects to attack plants in no choice tests because they have no choice. Some lepidopterans can even eat a paper if they are confined in a cage where there's no other food. So normally we don't take no choice tests seriously, but it is important that you do. So once you go to multi-choice tests, there we are trying to imitate nature, because in nature it's not just one plant, it's many plants out there. So the insects should be able to exercise its ability to choose, because that's what nature presents. Nature presents the choice. So if the insect is able to choose when it is given a group of plants, then that's a good sign that it is specific to those plants that have been chosen. I hope I answer your question.
Yeah. I just, since they can't complete development and it was just incidental feeding damage, I was just curious if the ones that did feed on Datura at all, if there was if they did any better on Datura than ones that didn't choose to, if you then, forced them to continue feeding on Datura or something. But it really just sounds like they're in an artificial situation and just kind of forced.
Yes. No choice is very artificial. Once you bring insects into multi-choice, that's where it becomes a little bit more natural, because this way they can somehow ignore plants that are not natural, and then focus on their natural host.
Awesome.
Thank you.
Okay.
Any other questions, anyone? I'm not seeing anything. Oh, yeah, okay. Here we've got one. So, hi, David, nice talk. You mentioned that different species of Cestrum have been eradicated. I remember having seen individuals of Cestrum elegans around Peter Maritzburg and Howick, maybe not in large numbers, but there are some.
Mhm.
What criteria do you consider to say that they have been eradicated? Thank you.
Yes, that's a very good question. As I said, we have an early detection program at SANPI. The person that asked the question, it looks like is a South African. Yes, we have the early detection program with SANPI. These are the people that are telling us that they no longer see these plants in the field, so they believe they have been contained. But it doesn't mean that they've stopped looking. They keep checking. But as I speak now, they cannot find any traces of these plants in the field. I think it has been 2 or 3 years. I don't know how many years. I'm not sure how many years it will be before they can really be 100% sure that they've been eradicated. But on the screen, I said contained because we don't believe that they may be totally eradicated. If I use the term eradicated during my talk, maybe that was a slip of the tongue. They've been contained. Thank you.
Cool. All right. Any other questions? I'm not seeing any. So you said you're hoping to reach out to people through the Solanaceae community and find additional collaborators, right, David?
Yes, that's true. People that are in South America, Brazil, Argentina, and Paraguay, all those places, if they come across Cestrum, any species of Cestrum, it doesn't really matter. If you see an insect, take a picture for me, or plants that are diseased with a pathogen. It can be a fungus, it can be anything, bacteria. Just take a picture and the location of the place where you took them and send it to us. We will highly appreciate that. But otherwise, if there's something that you want to know about the biocontrol on Cestrum, my email, I think it will be shared among the groups.
Yeah, I can definitely do that and send it. Just make sure your email's out there. So anybody who's watching or who watches the talk after it's recorded, and if you are interested in reaching out to David about your Cestrum observations, we'll hopefully can facilitate that. Yeah, so thank you again for presenting, and it's interesting to hear about that aspect of what's going on with Solanaceae. Yeah, and yeah, of course, you're welcome to join us for future seminars. They should be roughly at the same time, barring the time changes that are constantly making everything confusing.
Yes, I would be happy to join. Yes.
Great. All right. So before everyone goes, I'll just announce for our next seminar will be Edgardo Sepulveda, and he's from the CCC Centro the Research Center in Ensenada, Mexico, and he'll be talking about rhizobacteria associated with certain Solanum species and their effects on fusarium growth in tomato. So anyway, hopefully you can join us for the next seminar as well. And unless there's any last questions or comments, we'll call it a day. All right. Thank you, David.
Thanks a lot. Thank you.
Bye-bye.
Bye-bye. Thanks.